Thank you everyone for attending. I'm Qianru Hui, a PhD student from the Department of Animal Science, University of Manitoba. It's my honor to be here to share my study. Well, today I will be talking about the role of chicken calcium sensing receptor and calcium homeostasis in lean hens. I will be going through 4 parts to introduce the importance of chicken calcium sensing receptor in calcium homeostasis and its modulation mechanism. Then I will discuss the potential application of chicken CSR modulation on bone health and egg shell quality in lean hens. Finally, I will talk about the future work. Well, laying hens have a high demand for calcium to form eggshells during the active egg-laying period. Calcium is the most important mineral in the structure of bones and eggshells. In laying hen, approximately 2.4 grams of calcium is required in about 20 hours to produce a shelled egg. Only 40 to 75% of actual calcium can be provided by the feed via intestinal absorption, and the remainder must be mobilized from medullary bone storage. Well, the formation of medullary bone is always at the cost of structural bone, and the progressive structural bone loss causes osteoporosis, which contributes to the weaker and brittle bone and more susceptible to fracture if it's subjected to trauma, which are associated with pain, reduced egg production, reduced egg shell quality, food safety, and animal welfare issues. Also, eggs with good quality of shells ensure the income of egg producers and meet the demand of processing plants, egg traders, and consumers. And the egg industry also needs better methods that can be practiced by various sectors of this industry to mitigate osteoporosis. While chicken CSR plays a central role in calcium homeostasis, and there is a crosstalk between the CSR and vitamin D3 system in improving bone health and egg shell quality in lean hens. Calcium homeostasis is regulated through feedback mechanisms involving 3 hormones: parathyroid hormone, active vitamin D3, and calcitonin. And it is achieved by intestinal absorption, bone storage and resorption, and renal reabsorption. In addition to these pathways in lean hens, shell gland is a distinctive tissue to withdraw calcium for egg shell formation, which requires a high amount of calcium during egg laying stages. While CSR belongs to class C G protein coupled receptor, and it can sense subtle changes in extracellular calcium concentration and thus mediating pH secretion to maintain calcium homeostasis. In addition, calcium sensing receptor mediates a variety of physiological and biochemical processes such as ion channel activity, gene expression, inflammation, proliferation, differentiation, and apoptosis via inducing downstream signaling cascades. Well, why 1,20-half dihydroxy vitamin D3 is also important in calcium metabolism? Because a number of genes related to calcium transport and calcium metabolism has vitamin D response element located in their promoter region, and thereby inducing the transcription. These genes, including CSR and many calcium transporters, such as cobandin, calcium ion channels, and that's why active form of vitamin D3 facilitates the calcium absorption. Well, this figure shows that the relative mRNA balance of chicken CSR in the kidney, ileum, proventriculars, duodenum, and colon were significantly higher than that in other tissues at pig laying and lid laying. The relative mRNA balance of chicken vitamin D receptor was significantly higher in the whole small intestine when compared with other organs at three different laying stages. In the tibia, the relative mRNA balance of chicken CSR was significantly higher at late laying than at peak laying. As shown in this figure, the protein of chicken CSR and VDR was widely expressed in the kidney, proventriculars, gut, bone, and shell gland of laying hens at peak laying. While this figure shows the immunohistochemical results in the tibia, the CSR positivity was distributed within different laying stages, notably as circled in red. Medullary bone tissues were only observed in 40 and 55-week-old layers, which is because the medullary bone forms at the onset of sexual maturity. The positivity staining was mainly detected in structural bone, which is cortical bone, at 19 weeks, and mainly in the medullary bone at 40 weeks of age. However, at late laying, the positivity was found in both medullary bone and cortical. Well, according to the results, we concluded that chicken CSR and media are widely expressed in various tissues, including shell gland, but these two receptors distributed with different patterns. In addition, laying stages did not significantly affect their expression. In order to characterize chicken CSR and investigate its cellular modulation mechanism, we firstly constructed an in vitro expression system using HEK293 cell line, which stably expressed chicken CSR. The second step is to screen and characterize potential ligands using luciferase reporter system and also assays. GPCR transmits extracellular signals across the plasma membrane via intracellular coupling with G proteins. G proteins are classified as GIGQ-G12 based on their G-alpha subunits. Upon activation of CSR, G-protein alpha subunit dissociates from beta-gamma dimeric subunit, which in turn initiates a cascade of downstream second messenger pathways and eventually induces gene expression by various response elements. Well, figure 2ABC shows the effect of L-tryptophan on the EC50 of calcium. L-tryptophan induces chicken CSR activation required the presence of calcium, and the calcium concentration affected its potency. The EC50 curve for calcium was left shifted by 30%, 5%, and 1% in the presence of 1 millimole L-tryptophan via G-alpha-Q, G-beta-gamma, G-alpha-I, and G-alpha-12 mediated signaling pathways. Similar to human CSR, CMP production was inhibited after CSR activation via G-alpha-I mediated signaling pathway. In this figure, we found that the IC50 curve for calcium was redshifted by 10% in the presence of 1 millimole L-tryptophan. The chicken CSR response was significantly inhibited with increasing phosphate concentration by 92%, 31%, and 30% at 2 millimole phosphate compared with control in the three different signaling pathways. And when phosphate concentration was more than 5 millimole, chicken CSR response of G-alpha-I signaling pathway reflected by CMP level was significantly increased compared with 0.9 millimole phosphate, which is control group, and it was increased by 30% from 0.2 2 millimole more phosphate, and by 51% from 0.9 to 5 millimole phosphate. And from this study, we found similar to human CSR, L-tryptophan and inorganic phosphate were also chicken CSR modulators, acting as positive and negative allosteric modulators, respectively, despite agonism and antagonism in vitro. Both L-tryptophan and phosphate bind to the GFRQ-mediated signaling pathway. The results also provided theoretical evidence that L-tryptophan and inorganic phosphate could be used as potential nutritional ligands to prevent or attenuate chicken osteoporosis through chicken CSR modulation. Well, now we know chicken CSR is worth studying to mediate bone-related diseases in lean hens. Therefore, in the future, we will focus on CSR and investigate if the effective CSR nutritional ligands are able to improve bone health and axial collagen in vivo. After that, we'll explore the mechanism of chicken CSR mediating osteogenesis, which can regulate the formation of structural bone throughout the whole lean period. At last, we really appreciate the support from UNSERC, University of Manitoba, Manitoba Egg Farmers, and Wallenstein Feed and Supply Company. Well, thank you.
Thank you, Qianru. Again, I'll just remind you questions in the question and answer box. So there's one question here. It says, the effect of phosphate on receptor activation appears to be linear. Is it specific effect on calcium or receptor, or is it non-specific?
Well, for this question, I should be clarified because phosphate will, interact with calcium. Yeah, so we cannot do the direct the EC50 or IC50 curve because of this interaction. So we use one calcium mimetic, NPS R568, to simulate the function of calcium, and then we can just see this level and how phosphate can just inhibit the CSR to what kind of degree. That's why, we cannot perform the EC50 or IC50 for phosphate. So I think if you say it's not to be linear, just because of the experimental design. Thank you.